Hello and welcome to the respiration topic as part of the Unit 1 of the Higher Human Biology course. The process of aerobic respiration is split into three main stages, the substrates and products of which you need to know, the interactions, what's produced, what's used up, etc. So I'm going to take you through each of those three stages, first of all by naming it, telling you where it takes place, and then talking about the main stages of each. I'll put each one individually on the board, which means that you can watch this video in three parts, as and when required. So the first stage of aerobic respiration is called glycolysis. Glycolysis takes place in the cytoplasm, and it involves the breakdown of glucose into pyruvate. Glucose being a 6-carbon molecule and pyruvate being a 3-carbon molecule. Now, that's something that we knew from National 5. What we now understand is actually this process itself is made up of two stages. The first stage involves the phosphorylation, or the addition of phosphate to this glucose, forming us some phosphorylated intermediates. That word intermediates, if you remember, was used in the metabolic pathways topic as well, just to describe chemicals in the middle of a pathway. So glucose is first phosphorylated to give us these intermediates, and then this phosphorylated intermediates are then broken down into pyruvate. These stages have particular things about them. First of all, they have names, and they are named based on whether or not they need or release energy. The first stage is called the energy investment phase, and that is because 2 molecules of ATP are required. Now, when 2 molecules of ATP are used, what happens is this terminal, what we call the terminal phosphate, the end phosphate from ATP, is removed. That phosphate is then used to phosphorylate the glucose. What that leaves behind is 2 ADP. Now, we don't really need to worry too much about ADP, but it's worth mentioning that there is an interaction between this molecule, which is 3 phosphates, and that molecule, which is 2 phosphates. Having the phosphate added or removed is usually when energy is required or released. So we have energy investment phase. The second phase is called the energy payoff phase, and the reason for that is because 4 ATP are actually released as a result of phosphorylated intermediates being broken down. So what we say that occurs here is a net gain of 2 ATP. Okay, so because 2 has been invested, 4 have been removed or released. We have a net gain of 2 ATP, and that goes along with what we knew at National 5 level. The glucose is broken down to pyruvate, and that process releases 2 ATP. We know a little bit more now, but there's actually 2 phases, one of them which uses 2 ATP, and one of them which releases 4 ATP. The only other thing to mention at this point in time is that when phosphorylated intermediates are broken down into pyruvate, one of the processes that occurs is that hydrogen is removed. Hydrogen is removed with the help of a dehydrogenase enzyme, and the hydrogen carrier, NAD, is used. So NAD is converted into NADH with the help of this dehydrogenase enzyme, and so that's one of 3 places where you will see this happen, and that becomes very, very important when we get on to the third stage. So that's the first stage of aerobic respiration, glycolysis. Feel free to wait until you've got your head around that before you move on to the second stage. So, stage 2. Stage 2 is called the citric acid cycle. and that takes place in the matrix of the mitochondria. So just to explain where the matrix is, there's your mitochondria. This is the inner membrane of the mitochondria. That space in the middle that I'm shading in here, that's the matrix. Okay. Now, stage 2 starts with the product from stage 1, which is pyruvate. As a reminder, pyruvate is a 3-carbon molecule. The first thing that happens is pyruvate is broken down into a 2-carbon molecule called acetyl. Now, in that happening, two things occur. First of all, we produce or we release a molecule of CO2, which explains why we breathe out CO2 when we breathe. Another thing that happens is the same thing that happened in the first stage when a hydrogen was released. So a hydrogen is released with the help of dehydrogenase. This hydrogen carrier NAD is converted to NADH, and again we'll talk about what happens to that NADH when we get to the third stage. It's actually quite hard to see, so just to explain this, that says NAD, removed by dehydrogenase, hydrogen is removed by dehydrogenase to convert NAD to NADH. Okay, right, so now on to what the acetyl does. Now ultimately what the acetyl is going to try and do is it's going to try and combine with a 4-carbon molecule called oxaloacetate. Oxaloacetate combined with acetyl to form a 6-carbon molecule called citrate. But it's not quite as simple as that, because acetyl can't actually do that by itself. Instead, what it has to do is join to a coenzyme called CoA. Acetyl joins to CoA, much the same as hydrogen joins to NAD actually. CoA joins onto acetyl and allows acetyl to join with oxaloacetate. Because it's an enzyme, of course, though, it comes out of the process completely unchanged. So we have to understand that there's an interchange between acetyl and it joins to CoA to form acetyl-CoA briefly, and then CoA will drop acetyl off again once it's bound to oxaloacetate. So just to draw back on what we have talked about in other topics, what CoA probably does is creates an orientation state which allows acetyl and oxaloacetate to bind, whereas normally they wouldn't do that. So that's what CoA's job is. This, of course, is a cycle, so judging by the name, so eventually citrate is broken back down into oxaloacetate. Obviously, for a 6-carbon molecule to break down into a 4-carbon molecule, it loses a couple more CO2s, it loses a couple more carbons, which is obvious, but it does that in the form of CO2, so that's another further explanation as to why we breathe out carbon dioxide as a waste product. However, another thing that's produced is citrate. When citrate is broken down to oxaloacetate, is a little bit of ATP is released. So there would be a bit of energy released, so ATP would be produced on using that energy. And finally, this is the third and final place where NAD is reduced to NADH. We use that word reduced to mean it gains a hydrogen. So NAD is reduced to NADH, and again using that enzyme dehydrogenase. So that's the third and final point in time where we release NADH, and the NADH becomes relevant, very relevant when it comes to the third stage, which we will come to very shortly. So again, just have a wee think about that, go back through that in your own head, potentially rewind it to the start of this until you know stage 2 you're quite happy with, and then you can move on to stage 3. Right, stage 3 is called the electron transport chain. Among other things that has to be said, but that's as good a line as any, electron transport chain, and that takes place on the inner membrane of the mitochondria. Okay, so again, just to remind you, here's the outer membrane, here is the inner membrane. So all along this membrane, with what would it be, a membrane made of phospholipids, etc., and proteins. These proteins, a few of them that we'll talk about at the moment. So this line, I'll not draw every single phospholipid in, but I'm just trying to illustrate this is the membrane, the inner membrane. So this would be the matrix. I'll just put that there, matrix. And that would be outside the inner membrane, so just in this space here. So that space there, same as that space there. Okay, so there's two basically most important proteins found in the inner membrane of the mitochondria from our perspective, and that is hydrogen pump. So you get a few of them. And then there's another molecule called ATP synthase. And as you might imagine by the name, it's involved in the synthesis of ATP. So far we haven't really explained the production of all that much ATP. That's because the majority of it happens on the inner membrane of the mitochondria in that third stage. So we're now going to try to explain what happens to all this NADH that's being produced. You'll remember from the first two stages there are three different places where NADH has been generated because of the release of hydrogen with the help of dehydrogenase. Basically anywhere anything's broken down into something smaller. So the breakdown of phosphorylated intermediates into pyruvate, the breakdown of pyruvate into acetyl, and the breakdown of citrate back into oxaloacetate. All those are the three processes that release NADH. So what happens as NAD drops hydrogen off at this stage? The analogy I always like to use about NAD is it's like a taxi. So it picks hydrogen up, but a taxi crucially also drops things off. So it's dropped the hydrogen off. So we have a hydrogen, or lots of hydrogen, starts to appear near the inner membrane of the mitochondria. And what happens is it splits into two things. It splits into a hydrogen ion, and it splits into its electron. Both of those parts of the hydrogen atom that split have different roles. First of all, we'll deal with the electron. What happens is the electron is passed between hydrogen pumps, and that creates or gives the hydrogen energy. It gives it power to pump the hydrogen, and the hydrogen is then therefore pumped out of the matrix into this space outside the inner membrane. So lots and lots of hydrogen gets pumped out because of these electrons that are being used to provide the power. So what ends up happening is you are creating a very, very high concentration of hydrogen ions outside the matrix. So just on the other side of in this area here, on the other side of the inner membrane. That's where ATP synthase comes in, because hydrogen then just diffuses back through ATP synthase. Diffuses, of course, because there's been a high concentration of hydrogen created outside the membrane, so it diffuses back through, and as it does that, that generates lots and lots and lots of ATP molecules, and that's really where the majority of the ATP is produced in the process. So it's produced as a result of ATP synthase turning because hydrogen ions are moving by diffusion through it, and the only reason they're able to move by diffusion is because a very, very high concentration outside has been created by hydrogen pumps, which have been powered by the electron. Now eventually the electron gets to the end of those pumps, and the hydrogen, as it's passed through, these two things recombine to give us our hydrogen atom again. Now hydrogen needs to be dealt with in the same way as NAD needed to carry it. You can't just leave it floating about in the body. So what happens is we have what's called the final hydrogen acceptor, which binds to it. Final hydrogen acceptor, which is oxygen, which is why we breathe oxygen in. It's one of the main reasons why, well, the reason why we need oxygen. Oxygen is brought in, it combines with hydrogen to form water, and that is the final waste product. And that is the third stage of aerobic respiration electron transport chain. Again, I've gone through that quite quickly, so please make sure that you take your time to go through each individual stage before you move on to the next one.